Thank you for joining us for the fourth annual Global Summit on Kidney Innovation, Global Kidney Patient Voice, the key to accelerating innovation, a collaborative partnership event presented by the American Association of Kidney Patients and the George Washington University School of Medicine and Health Sciences. My name is Ed Hickey, and I am Vice President of the American Association of Kidney Patients and Chair of the AAKP Veterans Health Initiative. I'm a former Marine Infantry officer and am a practicing attorney in Los Angeles. I'm active in the Armed Forces legal community here in Los Angeles and have been work with the Veterans Legal Service Project of the LA County Bar Association, working to identify and address opportunities to assist veterans, active duty military personnel, and reservists with their legal needs. As a kidney patient and an experienced caregiver to those in my family who have also suffered from this condition, I am well aware of the need for innovation in the kidney care space. I've advocated on Capitol Hill on behalf of patients for innovations and worked with executive branch agencies to assess the efficacy of new methods for treating kidney disease. As an AAKP member, you can do the same. It is critical now that we as patient consumers raise our voices and create the demand for new diagnostics, devices, and biologics in our independent countries. Through AAKP Global, launched in 2019, AAKP is engaged internationally because we are committed to educating patients and leaders around the world on the importance of kidney disease prevention, innovations in treatment, and the opportunity to save more lives. We believe that a united patient voice can drive innovation and policy decisions that determine timely regulation and payment for new and safe care treatments. Globally, we are working with patients, medical professionals, researchers, and medical industry allies to cultivate a united consortium led by patients. AAKP believes this international consortium can advance innovation and change kidney care in countries where healthcare systems and/or infrastructure do not support patient consumer choice and/or access to care or treatments. For this session, titled Global Kidney Patient Voice, the key to accelerating innovation, government agencies prioritize global kidney health, United States, I am pleased to welcome an esteemed panel of speakers. To begin, I'd like to welcome a longtime friend and ally of AAKP's, Dr. Robert Starr. Dr. Robert Starr is Director of the Division of Kidney, Urologic, and Hematologic Diseases and Section Chief of Renal Diagnostics and Therapeutics Unit, Kidney Diseases Branch at the National Institutes of Diabetes and Digestive and Kidney Diseases at the National Institutes of Health. We're honored to have Dr. Starr with us to provide updates from the NIH through his presentation, Kidney Precision Medicine, How We Get There. Dr. Starr, the floor is yours.
We are out of focus, but Mr. Hickey, thank you much, and I'm really grateful to be talking. This was in focus, now isn't. But I'd like to talk about are a couple of things, mainly where are we, which is Innovation 1.0, and how this fits into the decade of the kidney from AAKP. And then I'd like to spend some time on where we are headed, which is really Innovation 2.0, and where we need your help. So this is about how we develop together a kidney precision medicine program, not just at NIH, but throughout the country and throughout the world. And what are the steps to getting there? So the mission of NIH is to understand fundamental knowledge of how the body works, but more importantly, apply that knowledge to extend healthy life and reduce the burdens of illness and disability. So it's acquire knowledge and then use that knowledge to promote health. What I want to talk to you about today is the intersection of those two and what we're doing and what we're planning to do. When we talk to patients, and the patient voice is really critical in our studies, patients have four questions. What do I have? What will happen to me? What can I do about it for me? And what does it mean for my family? This roughly translates in the medical sphere to diagnosis, prognosis, therapy, and genetics. Imagine a world if the healthcare strategies could be actually customized for every single individual patient. If you have a purple disease, you get the purple drug. If you have the teal disease, you get the teal drug. And if you have the orange disease, you get the orange drug. This is a customized precision medicine approach where the right intervention is given for the right patient at the right time. Talk about it. I'm going to take an example of urology just to show briefly how it works, then go back to kidney, I promise. So in urinary incontinence, there are 5 different types: dry, stress, where you cough and you pee; an urge, where you have to go to the bathroom quickly; a mixed type; and a 4th small cluster. What the LEARN network did, funded by NIDDK, Lower Urinary Tract Dysfunction Network, was to recluster the patients into more uniform groups of patients. These are radar plots, so the individual symptom scores, and the 4 new clusters are avoiding cluster, an urgency cluster, a kind of mixed cluster, and a cluster where everyone has all of the above. What the group is now doing is taking these clusters and figuring out, which is difficult, the pathophysiology under each cluster, and then trying to devise treatments for each cluster individually, so that we're not giving the avoiding patients the urgency treatment. What we have done, what we're working on now, is something called the Kidney Precision Medicine Project, where we do that kind of clustering, but instead of on symptoms, we do it on what we find in the kidney tissue. So we get research kidney biopsies from participants who are altruistically giving up small pieces of their kidney for research. We interrogate these tissues with a whole variety of very fancy technologies, analyze what we get, and look for targets for new therapies, and create a kidney tissue atlas that's given to the community free and is open, so that the whole community can now see what is new, what are new pathways, new potential treatment pathways. The whole purpose here is to improve the pipeline of drugs. This is an example from a study in patients who had a kidney transplant, where the investigators took tissue kidney biopsies from, I think there were about 30 or 40, 50 people in the study, and they took biopsies at 4 times before transplant and after transplant, and they ground the kidney up. It looked at the signatures within the ground up kidney tissue, and what they can see is this on the pseudotime diagram. They can separate out some groups that did quite well, that recovered. Some patients did quite well, and other patients developed chronic allograft dysfunction, and the signatures within the kidney tissue were quite different. Each dot is a person here. What we used to do, meaning 10 years ago, and frankly is still going on right now, is we take a look at the kidney tissue and we look at pathology of it, and we ask what cells are present, what patterns of injury are present. And we ask, how is the tissue responding? Does it look healthy? Does it look like it's undergoing good repair, or is it maladaptive repair that's going to cause problems? This is at the tissue level. What KPMP is going to do is look one step further and ask, what are the cells doing? What are the cells thinking? So the new pathophysiology is to look at the individual cells. Now each cell is a dot, and there are hundreds of thousands of cells, or tens of thousands of cells from each patient. And what this shows is a trajectory of what's happening to the thick ascending limb, TAL, that's part of the kidney, two different parts of it. As the cells are healthy on the left side, and they move to a more injured state on the right side, but it's an adaptive repair. The cells are actually doing something. What this analysis tells us is what drives the cells, the individual cells, to move from a healthy state to an adaptive state, or a healthy state to a maladaptive state. And more importantly then, what can we do to modify them? So this is the whole idea behind the Kidney Precision Project. Not just look at the kidney tissue by histology, but look at the cells and use a whole variety of different techniques to ask more detailed questions about what each cell is thinking, and how can we fix the cells, which will then drive what's going on in the kidney. So as an example, this is an example published by KPMP, where they had a 66-year-old woman who had the usual triad of hypertension, diabetes, and cardiovascular disease, whose protein was worsening in the urine, underwent our kidney research biopsy, and was discovered, no surprise, to have hypertension, some fibrosis, but no evidence of diabetes by light microscopy. However, when they dug a little deeper, they discovered that this patient also had completely unexpected mild kidney injury. When they went to the fancy single cell and omics kinds of techniques, what they discovered was, and there's a lot of information here, but basically they saw evidence of diabetes throughout the entire kidney at the molecular level. It was not seen at the light level, what would have been seen by standard pathology read, but the KPMP read was actually quite different. The patient had diabetic kidney disease and acute kidney injury. This completely changed the way the patient should be managed, change to an anti-diabetic strategy, and avoid things that might promote acute kidney injury. So that shows in one quick snapshot the idea of KPMP and what we're hoping to see that we don't see right now, what's sort of under the hood, what the tissue cells and pathways are telling us. Okay, so this is the next step, and this is the part that I have not shown in public before, but I'd like to present it here. And the next step is to do what has been done in cancer before, what the Neptune Match trial is also trying to do for glomerular disease, but now extend this to acute and chronic kidney disease, to basically all of kidney disease, and take the results of these molecular kidney boards that I talked about in the previous slide and turn it into a trial, a match trial, where you set up an infrastructure, where you recruit patients who undergo a now clinically indicated kidney biopsy. You use all the fancy techniques to test what their cells are thinking, to look for pathways that are amenable to treatment within the cells, and then figure out what treatment would be best for each person, match the participants to the particular trials that are out there, and then ask the question, does this new approach work better than just lumping everybody who has diabetic kidney disease to get the same treatment? We will start with repurposed drugs, but the whole purpose of showing this here is what we'd really like to do is get pharma involved and do this as a public-private partnership, a much bigger study that we could be not just testing old drugs, but also test new drugs that are specially formulated for working on individual pathways and also coming up with particular markers or biomarkers that tell you how to match patients to trials and also how to follow patients quickly to see if the drug is beginning to work before we see a change in creatinine, which may take months or years. So this is a really novel approach, a completely changed approach, a personalized approach to kidney medicine. And it really is where we think the future is going. What I've shown you are the steps that we're doing right now and what we intend to do over the next 5 to 10 years to begin to build a whole new infrastructure for how to do kidney clinical trials. And we hope that all of you will help us. We need your help. We need the patients, we need the participants, and we need pharma and academia to help us in this project. I thank you, and we'll take questions.
Thank you, Dr. Starr, for that wonderful presentation. And follow-up, I have some questions for you. As a doctor, scientist, and senior American civil servant, you've been at the forefront of involving patients in NIH research related to APO1, the Kidney Precision Medicine Project, and other efforts. Can you share with us how patients as collaborators in research have impacted your professional views and those of your colleagues?
So what we have learned to do, and in retrospect it's fairly simple, is to involve patients from the very beginning and at every stage of our studies. We have them at the first workshop. We ask them to tell us and the whole group why we are here, what is the problem we're trying to solve. We listen to them, we listen carefully. On one occasion I can think of an acute kidney injury study, we completely changed our approach from what we thought was going to be a drug study into a better treatment study, non-pharmacologic treatment study. We've used this in all of our studies, in several different transplant studies, including Apollo, including Think Your Next, which is a Hep C study. Apollo is looking at the effects of APOL1 both on kidney transplant recipients and as well as some donors, and it's used throughout KPMP. The patients bring all of their insights of their disease to the table. They're also really smart people. They're lawyers. They're communications experts. Some of them are scientists, and we listen carefully to what they say, and we use their thoughts to completely reshape the projects that we're working on. The other place where they're really helpful is in implementation. They help all of us stay grounded and make sure that we stay on target to finish what we set out to do. It's really easy when you're doing research to get stuck in a rabbit hole where you find an interesting direction and you want to go chase that direction. What we've done is use the patients to keep us all on track and make sure that we stay to the original goals of the study.
Thank you, Dr. Starr. Now, a final question. Can you describe some of the care innovations that are underway at NIH that may not be well known, especially to patients outside of the United States?
In a minute or less, it sort of boils down to we have several basic science type consortia. Good Map that's looking at sort of what are the parts list inside normal human kidneys. We have Rebuilding a Kidney, a really important study that's asking, okay, you have kidney damage. What do you do? Do you try and rebuild it, or do you build a new artificial or bioartificial kidney that's either outside the body or inside the body? And how do you work through some of the engineering challenges in terms of a vascular hookup and a plumbing hookup? There are all sorts of really interesting basic science problems there that translate into huge impediments to getting things, products on the market. And then we have several clinical trials, as I've outlined a bunch in transplant. One starting an acute kidney injury, a COPE-AKI, which I referenced, where a patient helped us decide that we should be studying. The real problem was that when patients left the hospital, they were left with no information, no idea they had AKI, no steps going forward. So this was to sort of make a cocoon around the patient to help them understand what to do, to give them a nurse navigator, a pharmacist to come to their house to clean out the medicines that they have in their medicine chest that are injuring their kidneys, et cetera, and to test whether this new approach is better than current standard therapy. So lots going on. That's the minute and a half answer.
There you go. Well, I wish we had a little more time, but thank you very much for your great insights, Dr. Starr. We appreciate you sharing your time and the NIH updates. Thank you very much.
Thank you.